So hi everyone, my name is Camille Trinidad and I'm part of the SNEV Journal Club team. SNEV is a virtual network run by students in the extracellular vesicle field with a goal to connect scientists and EV research students internationally. I just want to say thanks to Ken Whitwer for letting us moderate this session today. And for today, we have Liliia Paniushkina presenting. She obtained her master's degree in medical cybernetics and informatics at the Penza State University in 2017. She is a former early stage researcher at the Train EV project, granted by Marie Curie Innovative Training Network Program. She is in her last year of her PhD training at the University Hospital of Freiburg in Germany. She studies the role of oncogenes and their splice variants recruited to different EV populations in prostate cancer progression using patient samples and 3D cell culture models. Liliia will be discussing two papers with the overall theme of nucleic acids being presented on the surface of either cells or EVs. Feel free to type your questions in the chat box. And with that, Liliia, can you please share your screen and start your presentation?
Here we go, right?
Yes, I can see it. You can start.
So perfect. Thank you so much for letting me present on this EV Journal Club today. Without any further delay, I will start from the content. So today's presentation is going to have two parts. In the beginning, we will be talking about small RNA modified with the N glycans and displayed on the surface of living cells. And in the second part, we will be focused on antibiotic-induced release of small extracellular vesicles with the surface associated DNA. So let's go from the glycoRNA part. So until now, the glycans were known to regulate the different cellular functions, such as the proteins and the lipids trafficking, embryogenesis. They've been involved in the host pathogen recognition, and they also play an important role in tumor-immune interactions. So the authors of these publications, which have been published recently in Cell, aim to understand whether or not there is a linkage between the RNA and the glycans. So they use a very interesting approach. So they use the glycan metabolic reporter. So for the cells and the mice model, it is tetraacylated mannose, which contains the sugar part and azide. It's normally metabolically naturally incorporated to the cellular glycans. And then from those cells, the authors isolated RNA. So the RNA isolation been done using the different steps. So first of all, they applied the triazole, the highly concentrated salts at the 37 degrees, and they washed it out by the column to have the mixture of the RNA, DNA, proteins, and other components. So using the proteinase kit treatment, they get rid of the proteins and clean it with the column again. And afterwards, they applied the quick reaction with the azide using DB's OPG4 biotin. So this molecule immediately binds to the azide, and in the 50% formamide at 55 Celsius degrees, it also reduced the DNA, and it's making the complex, which is contained the biotin and could be further visualized on the scanner. So the solution before the biotin conjugation, they were denaturated in the 95% formamide. And then using the formaldehyde agarose gel, the streptavidin reaction, as we all know, with tetramer and biotin conjugation was visualized using the scanner. So where we normally got the green fluorescent signal. Overall saying, if the sample contains RNA with incorporated cellular glycans, we should see the green fluorescent signal. So the result is shown on this slide. So they've been treating the HeLa cells every 6 hours, and after the 12 hours point, every 12 hours with this glycan metabolic reporter. And they observed the presence of the high molecular weight region band, which was appearing after 12 hours of the treatment. To actually be sure that it is really RNA structure on the picture you see here, they performed the, first of all, DNase treatment, which did not affect the signal and the band didn't disappear. However, the RNase treatment really confirmed the presence of the RNA structure. And then interestingly, the combination of the RNase together with RNase inhibitor also let visualize the band, which they call the glycoRNA. So showing this phenomenon on the cell culture model, the author has been wondering whether or not it's cell culture specific and could it actually be implied for the in vivo model. And indeed, so they injected intraperitoneally the mice with this glycan metabolic reporter every 2 and 4 and 6 days. They harvested the organs and isolated RNA from them. Here you see the RNA from the liver and spleen, and you could appreciate that there is a glycoRNA band distributed in a dose-dependent manner, and it also RNA is sensitive. So therefore, the authors confirmed that the glycoRNA is not an artifact of the tissue culture. And then there was a question about what the structure of those glycoRNAs. So the glycoRNAs got RNA and the glyco part. So let's start from the RNA. Seeing the behavior of those glycoRNAs in the gel, the authors hypothesized that it's more probably the large RNA fragments. However, when they performed the polyadenylate enrichment, they were not able to see any signal. So they changed the strategy and they done the size-based precipitation and observed the clear band in the area known for the small RNA appearing in the gel. So just to be really precise, the sample been loaded to this cross gradient from 50% to the 30%, and at the band area in the fractions where the glycoRNA signal still appeared, they visualized by the cyber gold staining the presence of the band at the small RNA area. So those fractions been taken and sequenced, and they were able to identify that they contain different types of RNA. For instance, yRNA, small nuclear RNA, ribosomal RNA, SNO RNA, and cRNA. So they've been also investigated the potential role in glycans in the yRNA using the CRISPR-Cas reaction, but for the time spare, I'm not showing this experiment here. So let's go to the glyco part of the glycoRNAs. So they suggested that the known celiac acids play a role in the cell-cell communication, and they mainly occur in the terminal components of the cell surface in the glycoproteins and glycolipids. So it was an idea to check whether sialic acids are present in those glycoRNA preparations. To avoid any sort of interference with the transporter, they've isolated the RNA from H9 cell line, and then they labeled by DNB reagent, which is fluorescent itself, and then loaded the samples to the high-performance liquid chromatograph. So using here in the blue, the mixture of sialic acids reference panel, they were able to visualize the different peaks, which characterize the different sialic acids form. So in the presence with no enzyme, the authors seen the signals at the RNA and two known U5GC and U5AC sialic acids. And they confirmed that by the celiac treatment and RNase cocktail treatment, those peaks were disappearing. So with that idea that sialic acids are actually components of the glycoRNA, the authors been investigated what is the localization of those glycoRNAs. So they taken the 293T cells, they obtain after the treatment separately the nuclear, cytoplasmic components, and the membranes. And interestingly, in the picture AB here, you can appreciate that there were no glycoRNA band appearing in the nuclear and at cytoplasmic area. However, the signal being at the membranal part. So as we know, the membrane got very defined topology, and the question whether it's on the luminal or external part of the membrane actually glycoRNA located. And they perform an experiment with the crude membrane isolation using detergent Tritonix, which is supposed to destroy the membrane, and VCC, which is the enzyme that digests the sialic acids. Then after that treatment, they've isolated RNA and again used the previously described DMB derivatization to show whether or not the peaks which are special for the nu5AC or nu5GC are present or not. So overall, this experiment showed surprisingly that it's not really important whether Triton X treatment happened or not for the sample. Anyway, it was a decrease of the sialic acids in the sample after enzyme digestion. However, some sort of the decrease been also observed after 0.1% Triton X treatment impact on the nu5 AC sialic acid, suggesting that indeed the glycoRNA localization is most probably on the membrane surface, but also could be inside. So to investigate this question further, they analyzed the cell cultures with enzyme treatment. And they observed that it was a glycoRNA reduction over like 50% of the signal after 20 minutes of the enzyme treatment, suggesting that indeed half of the glycoRNAs on the cellular surface. So they perform an experiment with the 3 different cell lines with the HeLa, 293T and He562. So the last one is actually in suspension to visualize that it is a significant decrease in the glycoRNA signal after digestion. They also applied the non-genetic strategy is related to the lectins binding. And if you're interested in that approach, I will encourage you to go to the method section of the paper. So to summarize, a glycometabolic reporter incorporates into the cellular RNA, which is true to the cellular and the mice model. GlycoRNAs are small non-coding RNA, and the glyco part of them is chelated. The glycoRNAs are associated with the cellular membrane, and they gain an access to the surface of the living cell. So when we're talking now about the surface of the living cell and the cellular membrane, it's the best time to go to the second part of the journal club and discuss the role of antibiotics induction on the release of small extracellular vesicles with the surface associated DNA. So in this study, the authors been exploring the effect of the ciprofloxacin on the cell culture and EVs released by them. Ciprofloxacin is a widely used antibiotics used for the treatment of bacterial infection. It suppresses the replication of the bacterial DNA, and it also used to treat and prevent the mycoplasma contamination, which is still, even though 50 years ago discovered, a widely known problem that some of the cell lines are contaminated, and it's very tiny organism that could be really hardly detected, so we're trying to avoid it. So the question is, how the ciprofloxacin exposure affects and whether it has any sort of an effect on the EVs. To answer this question, the authors used the 3 different cell lines: Burkitt cell line, human lymphoblastic QT cell leukemia harvested, U937, human monocytes from the histolytic lymphoma, and the 3rd line was from human pancreas carcinoma neopaka. So those cells been grown and cultured from 40 to 60 days with presence or the absence of the ciprofloxacin. Then, after the certain point, when they reached the 50%, they've been used for the EV production and the protocol that I'm explaining here. So the EV has been isolated by the differential centrifugation with combination of hydrostatic gravity filtration. So the vesicles been isolated by their size and called large, medium size, and the small EVs. Additionally, small vesicles been purified using optiprop density gradient. So of course, the first thing that been performed, I'm not showing it here, whether or not ciprofloxacin exposure got an effect on the cell viability and apoptosis. So there were no observed effect. And using the flow cytometry for the small vesicles with latex beads, the authors showed that first of all, there were no change in annexin 5 signal under ciprofloxacin treatment. Also, there were no change in CD63, suggesting that ciprofloxacin treatment didn't affect, in this case, EV amount. But surprisingly, the staining with the propedium iodide, which stains DNA, and anti-H2B, like the histone 2B, they were able to observe the shift of the signal to the positive side under ciprofloxacin treatment, which wasn't the case for the large or medium-sized vesicles. When in the large vesicles, it was only a positive population in annexin 5 staining EVs, and large EVs were absolutely negative to the histone. So the same effect being observed for the pancreatic carcinoma neopaka cell line. For the small vesicles, it was an additional peak in the propedium iodide staining, but there were no effect for the U937 cells. So seeing this phenomenon, of course, the question was, is it due to the presence of the DNA in the small EV samples? And the authors isolated DNA from the large, medium, and the small EV populations. And with the usage of the DNA's one digestion, they've been able to observe the significant decrease in the DM amount in the small vesicular sample. And to confirm that the DNA's treatment didn't affect the amount of the small vesicles, they were staining the vesicles with annexin 5 and CD63. There were no change in the positive population. So, is DNA associated with the small EV surface? Starting to answer this question, it's nice to know whether the PI and CD63 are actually co-localized or not. The authors applied the optiprop density gradient for the small EV population, keeping in mind that DNA classically in the density gradient appears in the fraction in 1.21 to 122 gram per ml. They analyzed all the fractions in the presence of CD63. So the fractions 6 and 7 were CD63 positive. And also I'm showing here the data on the PI staining, so that the fraction 7 at 109 gram per ml density was highly PI positive, while the fraction 6 was not. So the small EV fraction at 109 gram per ml density was CD63 and propydium iodide positive, suggestion that it might be a co-localization of DNA in small vesicles. So the authors also used the high cell concentration to release the DNA from the small vesicles. They observed that in 2 molar natrium chloride concentrated solution reduced propydium iodide signal in the small EV population, which wasn't the case for annexin 5 and CD63 signal. The control with latex-bound DNA didn't show any difference. And from there, they concluded that the high cell concentration releases DNA from small EVs rather than EVs from the beads, and the DNA might be associated with the small EV surface. So next, they were investigating the effect of this phenomenon on the adhesion properties of the vesicles. And they applied a very interesting approach. So they used the resonant waveguide grating base label free optical biosensor. So what is that? In the 96 volt plate, it is a biosensor being placed on the bottom of the well. It's a tiny film covered by the niobium pentoxide, and it has known resonant wavelengths and also known refractive index. So which also means useful when the bimolecular adhesion happens. The camera could detect the shift in the resonant wavelengths and detect it, and then transfer, because of the liquid, to the refractive index changes. So in the picture B, you could see the experiment with the serum albumin and fibronectin coating of this biosensor, that when those two solutions been added, it made a stable line. So the coating did happen, and even after the PBS washing, the signal was still stable, which suggests that it was irreversible absorption of those two components. So coated the biosensor with the fibronectin and serum albumin. The authors were wondering what would happen if we're trying to analyze the large, medium, and the small EV populations with this biosensor. So you could appreciate here that during the time after 15 minutes, when the stable line been reached, in the large EV case, there were no observed increase of the refractive index and any changes for the large vesicles. However, the small EV sample been clearly showing that it is an increase in the refractive index, which suggested that binding did happen. To check whether this binding has something to do with the DNA, they applied the DNase 1 treatment and observed the significant decrease in the signal that the DNase 1 digestion in fibronectin-coated biosensor, saying that small EVs from the ciprofloxacin-induced uric cells bind to the fibronectin-coated biosensor. So to go in deeper to the potential reason of the DNA being on the surface of the vesicles, the authors used the uric cells under ciprofloxacin treatment with activation, with apoptosis, and with no induction, to actually isolate in large, medium, and small EVs, whether or not to see some sort of the changes during this physiological procedure. So they analyzed the particle size and number, annexin 5 pi distribution, DNA, and the proteins. So not surprisingly, the data from the Q-nano TRPS with the different membranes showed that apoptotic uric cells produce more large and medium-sized EVs with no difference between the control-activated and apoptotic samples. And activated uric cells tend to produce more small vesicles, which you could appreciate here in the green. So investigating the potential localization of the DNA on the surface was performed the staining with Tridonix to annexin 5. You can see here, so the authors claim that the annexin 5 signal for the control activated apoptotic cells was really high compared to the other vesicular subpopulation, which I personally don't really see because it's still high without Tridonix treatment. However, under the Tridonix 100 treatment, there was a significant reduction in the annexin 5 positive events in both cases. Interesting part that propedium iodide staining in the large vesicles didn't show like so much positive signal in the control and activated cases for the large EVs, but it was an increased outer suggestion of significant under the Tridonix treatment in the apoptotic case in the PI staining. However, it was sort of stable, and here the idea was that it could be due to the transglutaminases cross-link with the propedium iodide in the surface of the large vesicles. While in the case of the small EVs, the signals from either treated with the triton or not for all control activated and apoptotic samples didn't affect the positive PI stain populations, and we could confirm that here it's really detergent insensitive PI signal. So the authors also show that it is the presence of the chromosomal DNA only in the small vesicles, and its DNA is one digestion sensitive. However, all of these subsets contain mitochondrial DNA. And in the large EV sample, it wasn't sensitive to the DNase 1 digestion. However, in the small vesicles, it was for all three experiments. And additionally, with the mass spectrometry and then proteomics later on, the authors showed that the small EV protein samples contain histones and also mitochondrial DNA binding protein in case of the control and apoptosis sample in very high level. So together confirming that indeed, even in the proteomics analysis, apoptotic urcat cells were producing more specifically in large EV population apoptosis related proteins. Here you could appreciate that the small EV sample contain more specific and unique proteins out from activated urcat cells, but not from the apoptotic. So to sum it up, sustained exposure of the urcat and Miyapaka cells to the ciprofloxacin may induce the release of DNA associated with small EVs. Ciprofloxacin induces release of both the chromosomal and mitochondrial DNA associated with the EV surface, small EV surface. And the conclusion that the surface-bound DNA facilitates the small EV binding to extracellular matrix protein fibronectin. With that, I'll stop my talk, and thank you so much for your attention. I hope you've seen something interesting to you and that will lead us to the nice discussion. Thank you.
Thank you, Liliia, for that wonderful presentation. We'll move on to the questions. Let's see. So no one has any questions in the chat box yet, so feel free to type in your questions so that we can bring it up. So I guess I will start with a question that my question first. So in terms of treating cells with ciprofloxacin, how long is the phenotype for these cells maintained, or how long is it that they're giving out these EVs that have DNA on the surface. And then another question I have is like, what do you think could be the function of these DNA found on the surface of EVs, and what happens if it gets uptaken by other cells?
Okay, yeah, thank you for a question. So it's a long exposure. So the treatment was done, for instance, in this case, from 40 to up to 60 days, like 1 month. It's not a one dosage effect. So I guess it's different from cell line to cell line, but at least the 60 days, that was the maximum here. And of course, I don't want to speculate, but ciprofloxacin normally binds to the topoisomerase, DNA topoisomerase in the bacteria. And there were some publications suggesting that it might also have an effect on the cancer cells. So DNA on the surface of EVs, I don't know, maybe just a stress reaction. Whether or not it's got a function, really, it's hard to say. What it would be really interesting if the authors will go on and using this biosensor with different coating, with a different specific coating to the DNA, RNA, also taken together now the new knowledge about the glycoRNA maybe, and then we'll try to catch those vesicles and have a look, I mean, what's going on. And also, of course, the functional study is needed. So I can't really say what's the function, but for example, in the first publication, the idea was said that it increases the surface, the cellular surface, is sort of like improve the binding. Who knows, maybe for the vesicles case, it could also be not that specific, not as specific as we might think about that. So, but this is just my speculation.
So we do have another question. So thanks for that. Mad Husadan, could you please unmute yourself and ask the question that you have on the
Thank
chat
you.
box?
Excellent presentation and very nice summing it up with both DNA and RNA on the surface of the cell membrane and EVs. So I have a question regarding the first part about glycosylated RNA. Did the authors mention or do you know that how these glycosylated RNA, for example, why RNA are anchored to the surface, cell surface? Are they like anchored through some proteins or lipid interactions? Or?
Okay, yeah, thanks for your nice words and also the nice question. As far as I remember, no, it's really like the beginning to show that those glycoRNA actually exist. And I'm sure it's the next question to understand the mechanism why some RNAs became glycosylated and then go into the surface inside and outside instead of doing other useful things in the cell. So to my knowledge, I don't remember the answer to this question, but I'm sure it's going to be the next task for the authors, for sure. Yeah.
Yeah, definitely. Thank you.
Just to follow up with that question, do all glycans contain sialic acid?
No, I don't think so.
Okay. And so the next question is from Brooke Brown. Could you please unmute yourself?
Hi. Yeah, I have a question about if you have any idea of how much RNA you think would be incorporated, how many RNA molecules you think might be incorporated onto the surface of the EVs. And following off of that, do you think that, you know, what I've taken from the Carabal-Tosi paper, do you think any of the glycoRNA might be associating post secretion from the cell?
Okay, yeah, thanks for a question. I think it's all about what it's doing there. If it's non-coding RNA, it could be fragmented. If you're asking about the methods which the authors maybe used in the first paper, they were basically talking all the time about from 50 up to 20 microgram of the RNA. in the EV case, it always depends on the cell line. Yeah, I mean, in your question that do you think that the glycoRNA could be incorporated on the surface? Yeah, well, why not? If there is a space, if there is a space for again this non-coding RNA being glycosylated, so I don't know whether I answered your question or not.
Thank you.
So we have a comment from Phil Ascanes. They said that they're able to dose response add MIR150 to activated small EVs with in vivo bioactivity dependent on MIR150s. Phil, do you have anything to add to this, or do you have a question or comment?
I wish people would follow it up. It may be related, but we first found through cloning that an activated subset of T-cell derived EVs were associated naturally with microRNA 150. We were later then able to take 150 knockout EVs from 150 knockout and to add externally 150 in a dose response fashion. In one set of published experiments, as little as a femtomoles and dose response added to 10 to the 10 activated small EVs had then in vivo bioactivity. In another paper, it was nanomoles. And so we have not been able to distinguish whether this active MIR150 is on the surface. We're just adding it at 37 degrees or is inside. But it's certainly a potential claim that added surface 150 onto small EVs can lead to mediation of significant biological effects.
Really interesting, thank you. And do you have an idea whether it might be glycosylated if we're talking about glycoRNA right now?
Well, glycosylated just happened last week, so we haven't had time to look into that.
Yeah, thank you so much.
But I wish that others would try and follow this up. So we have an in vivo bioactivity that's dependent on 150 and not random MIR or reverse sequence, but on 150 that can be added to activated. Activated is very important. It's less than 10% of the total, and we isolate them from affinity columns that either have antigen, because they're antigen specific, or anti-CD9 on the columns, and that 10% activated has the ability to associate with MIR150 that has biological activity.
Yeah, I'm sure there are also other studies investigating the roles of the RNA, microRNA in EV delivery and the adhesion and so on. So my very naive idea was to also be in state of the art, and then maybe like start thinking about that these glycoRNAs could also be present on EV surface, and maybe metabolomics together with proteomics and other studies could really help us, and with the biosensor, because I think the biosensor from my point of view got very high potential, and to really see whether it's not is a surface associated. So I was wondering maybe somebody could share an experience also from their laboratories, what they're doing to understand where the RNA is, outside, inside, surface, transmembrane, and so on.
In our case, we are adding commercially produced MIR150 that is not glycosylated.
Okay.
Does anyone else have anything to add or any more questions?
Well, I think it's important in this kind of work to have biological function at the end of the rainbow.
Yes.
To just follow around molecules is very interesting, but I would urge people to work in a system where you have a biological function that's something that's can really be demonstrated with unambiguous assays to distinguish the biological significance of RNA on the surface or DNA on the surface or glycosylated nucleic acids.
Definitely, I agree with you, Phil. It would be interesting to find out like what happens, to follow it from like how it's being put on the surface or in EVs until how it's what happens after uptake. If the cells get uptaken, what if there's a change in the phenotype? That would be great. If we have like more technology showing up that would make it easier for us to trace that.
The fact that it's happening with activation, I think, is a strong hint that this is biologically significant.
So I think there's no more questions in the chat, and so we're gonna just end this early. Thanks everybody for your questions, and thank you for joining us today. Thanks again, Liliia. I hope you all have a great rest of your week. Bye.